All right, let's get started. People will get started. All right, so our first research talk is from Supratim. Supratim is at IISER Kolkata and has done a variety of interesting questions, some of which touch upon population genetics, but many of which touch upon broader themes in evolutionary biology. And so he's here. Do pick his brain later on about other things that he's done, including codon bias, which is why I know him. And so today's talk is going to be about RNA world and the origins of life. Supratim, happy to have you here.
Yeah, thanks to Deepa and Kavita for giving me the opportunity to present this work. So around 4 billion years ago, a remarkable series of incidents culminated in the origin of the first living organism. And before we try to address the question of how that came about, it's helpful to recollect what evolution looks like as we know it today. And this has already been touched on by some of the previous speakers. It's not changing. Oh, you can. Still not changing. Okay, so it's helpful to recall what we mean by evolution as we see it today. So there are certain characteristics that's good to go through. So we need probably better to use this machine rather than my machine. Okay, so we have heritable genetic information that is encoded in DNA sequences. Phenotypic traits are determined by genes that encode proteins, which assumes some sort of a genotype to phenotype mapping, which can be seen via a sequence of amino acids or nucleotides gives rise to the wing patterns on a peppered moth here. And random mutations produce variants on which selection acts. These variants sometimes are well adapted to the environment and they increase its frequency. But when the environment changes, other variants can take over, as is again seen in the transition of the peppered moth population from light-colored to dark-colored and then back to light-colored. And vertical descent, because individuals pass on their genetic information to their offspring. And that's how information is propagated from one generation to the other. And vertical descent also allows us to establish evolutionary relationships between different organisms that allows us to draw phylogenetic trees and hopefully possibly trace back the origin of all living organisms to the so-called last universal common ancestor of all organisms. So these are the characteristics that we find in evolution as we see it today. But 4 billion years ago, there were no genes, there were no proteins, none of the molecular machinery which is essential for replicating an individual organism existed. So how is it that life originated from a bunch of chemicals and eventually got to a stage where we have genetic information encoded in DNA and regulatory and catalytic tasks are performed by proteins? So this is the broad set of questions that I'd like to address in this talk. And many years ago, group in NASA was given the job of coming up with an appropriate definition for life. And they came up with this definition. Life is a self-sustained chemical system capable of undergoing Darwinian evolution. And even though this is not a perfect definition. There are certain drawbacks. It's a good working definition because it helps us to break down the question of origin of life into parts. For instance, we can ask, how did a bunch of chemicals come together to form a self-sustained chemical system? And the second part of the question would, how did that self-sustained chemical system transition to a stage where Darwinian evolution was possible? So we start from a bunch of chemicals which are nucleotides, and answering these questions would require us to understand how from a set of nucleotides we can end up with a functional biomolecule like an enzyme, and eventually how does these functional biomolecules get encapsulated into containers, cell-like containers like protocells, and then selection starts acting on these protocells because of variation in the contents of these protocells, as a result of which these can proliferate, and we want certain types of protocells to proliferate, and whether the ones that proliferate encode more functional diversity in terms of the kind of actions they can perform. And so we want to get from this stage to this stage, which is where evolution really sets in, because it can act on the different protocells and decide which ones can increase its frequency relative to the others in the population. So I'll be talking about a hypothesis which was made sometimes in the late 1960s called the RNA world hypothesis, which says that unlike the DNA protein world that we know of today, there was no division of labor in the primordial times before DNA and protein evolved. All the three functions of information storage, catalysis, and regulation were performed by RNA molecules. And evidence for that exists because RNA enzymes called ribozymes were discovered, and RNA regulators called riboswitches were discovered more recently, about 20 years ago, which have been found to regulate the concentration of small molecules in cells. So an RNA molecule can perform all the three functions that are performed individually by DNA and proteins in the current world today. So the hypothesis is that the earliest living organisms were made up of RNA molecules only. So even if this hypothesis is true, we need to understand how we can start with a bunch of nucleotides, RNA nucleotides, and because initially there were no enzymes to begin with, how did non-enzymatic chemical reactions produce these biopolymers, these long RNA chains? And more importantly, it's not enough to produce RNA chains. We need to produce functional RNA chains which can have certain functions like enzymatic activity. So how did RNA enzymes like ribozymes emerge through non-enzymatic reactions only? And eventually, if you are interested in answering the question of origin of life, we want to understand how these enzymes got encapsulated in compartments, and how did these compartments, how was variation introduced into these compartments, what were the processes involved, and how did selection start playing a role, choosing certain types of compartments over others. So from this point, we need to go to a living protocell, which is a self-sustained chemical system, and we'll see to what extent it can be subject to Darwinian evolution. So the first question that I want to address is, starting from a bunch of RNA nucleotides, how can non-enzymatic reactions produce something as complicated as a ribozyme, which has to be a long RNA, which has a complex folded structure, and so on? And what are the environmental conditions that were necessary to favor the emergence of such long functional biopolymers? And whether these different types of biopolymers which are produced as a result of non-enzymatic reactions can be classified based on their secondary structures. What do the secondary structures tell us about the functions that they can perform, and so on. So we also want to understand what impact the earliest ribozymes had on the protocellular evolution once they got encapsulated in these compartments, and the nature and hierarchy of these earliest ribozymes. So which was the most useful ribozyme to evolve first, and subsequently can we talk about a hierarchy of ribozymes of different functionality that arises inside these protocells, and then selection starts acting on these protocells. So how do a bunch of chemicals end up producing a long functional biomolecule like a ribozyme? So one mechanism is just by the process of concatenation, you add one monomer after another, and that produces a monomer chain, the polymer chain. But this process is slow, and these chains can be broken up into parts, so it's not good enough to produce a long polymer. But another mechanism which is found to be useful is called template-directed replication. And here you have a template of nucleotides already produced, and then nucleotides can add across the different monomer, the nucleotides that make up the template. So this is called a primer. The primer gets extended, and so a complementary copy of the original template is produced if there are no errors during the incorporation of nucleotides. So, so a new nucleotide can get added, and so on. So, but a primer extension is not without errors. So errors creep in. A wrong nucleotide gets attached here, leading to a mispairing, and that can affect the sequence of the primer that is produced from the template. So the primer that is produced as a result of error-prone replication process can have a sequence that is very different from the one that is complementary to the template. And it is initially this difference due to error-prone replication that plays a role in sequence diversity that was essential ingredient for these ribozymes and protocells to emerge. But there's another competing process as well, which can break up these polymer chains, and this process is due to hydrolysis, and these bonds can be broken up. But if existing, some bonds are better protected than others because of base pairing. So bonds in this region are completely unprotected, so they are more likely to be broken than bonds in this region, which are protected because of base pairing, and so on. So there are two competing processes at work here: template-directed primer extension, increase in polymer length by concatenation. Both lead to long polymers, whereas the antagonistic process is the hydrolysis process, which tends to break up these long polymers into shorter chains. And so there has to be a mechanism by which we can still produce long polymers which are functional despite the fact that they can be broken up. So that requires the rate of hydrolysis to be small compared to the rate at which polymers are increasing in length. So one way to do that is to look at possible environments in which these polymers first started forming. And one such environment that has been proposed is an environment near a hot spring, which undergoes this cycles between hot and dry and cold and wet phases. So this environmental cycling was shown to increase the average length of the polymers. And the reason is intuitively simple to understand. So if these two processes of hydrolysis and polymer extension through template-directed processes or concatenation processes take place simultaneously, then the polymers which form are also simultaneously being chopped up into small pieces because of hydrolysis. But if we can separate these two types of reactions into different phases, because hydrolysis requires water and it's more likely to happen in wet, cold conditions, whereas the polymer extension process is more favorable in dry or semi-dry conditions, then it is possible to produce long polymers which can eventually fold into complex structures and hopefully acquire certain functions. So this is exactly what we tried to do. So we divided a cycle of 24 hours into 3 phases: a morning phase, a morning to noon phase, which is a dry phase, which favors these concatenation reactions, an evening phase with a semi-dry phase, where these template-directed primer extension processes, which occur at higher rates, are favored, and a night phase, which leads to breakup, hydrolysis and breakup of these polymer chains. So we started with a population of just nucleotides and monomers, and we used rates which were derived from experiments to try and find out how we can, how long a polymer we could produce through these 2 processes of concatenation and template-directed polymer extension. And how that depends on the duration of the dry, semi-wet, and wet phases. So we found that if the duration of the dry and semi-wet phases are favorably tuned, polymers can grow quite long. The average length can be as large as 40 to 50, and the maximum length can be as much as 100 in favorable conditions, where the duration of the wet phase is of the shortest duration. So no, so the template-directed primer extension processes are favored in the semi-dry conditions. Those are the faster processes that are taking place. The concatenation process is slower, but you need both processes because you need a template size to also increase. Otherwise, if the template size is fixed, then the complement which is produced will also be of the same size. So you want the template size to keep growing, which is why you need the concatenation process, which is favored during the dry phase. But you also need the fast-acting primer extension process, which is template-directed primer extension process, which is why the semi-wet phase, a larger semi-wet phase favors the overall polymers which have longer length. So, yeah. So it's a competition between these. It's a competition between these 3 processes, as well as the duration of the 3 phases.
So can you, for example, collapse these curves into one with these 2 parameters?
With there are more than 2 parameters. There's a there are 2 T's. You cannot, you can have a same duration of all the 3 phases, which is not here, which is here, which is this green curve here, because so 888, and if you have the same duration of the 3 phases, you will get lengths which are less than 30, average lengths less than 30. And you can see it from here also. This is the distribution of the maximum length averaged over several days. And as the duration of the dry and semi-wet phase is changed, so when the duration of all 3 phases are equal, the maximum length that you get is of the order of 64. So the reason the length is important is the earliest ribozyme, for instance, the hammerhead ribozyme, which is capable of cleaving itself, it is found to be of the order of 50 nucleotides. So you need sequences which are roughly more than 50 nucleotides for biological function, useful biological function to be encoded in them. And so the larger the sequences you can produce through this non-enzymatic pathway, the more likely you are to end up with functional biopolymers which can act as ribozymes. Okay, so that's the idea. Okay, so we generated a lot of these sequences through these processes of template-directed primer extension and concatenation, along with the competing process of hydrolysis, and looked at the kind of secondary structures that were obtained when you fold these sequences. And so initially, for small sequences, very simple secondary structures were produced, so they were most likely not functional. But what we found to our surprise, that when you go to longer sequences, the secondary structures produced are somewhat similar to what is found in nature. So for instance, this is a structure of a hammerhead ribozyme, and this has a tRNA-like structure for those who are familiar with tRNA sequences, the secondary structure of tRNAs. And so these kind of structures were generated when we started folding these sequences that were produced due to these three processes. So the conclusion of the first part of the talk is that long structurally complex RNA sequences can indeed be produced through non-enzymatic reactions, but you need favorable environmental conditions like environmental cycling between hot and dry, semi-dry, and wet phases. And you need both concatenation and template-directed primer extension processes to go on in order to counterbalance the effect of hydrolysis, which tends to break up these long sequences. Yeah.
So the heat map is showing percentage of the structure that you've shown as an example?
Yes, yeah.
So maximum you get of these complex ones is 1 or 2%.
Yeah, yeah.
And so there are other structures, but they're just...
they are not very useful structures. So they are
quite low.
Yeah, it's true. Yeah, but if you have a lot of these reactions in a given region, then that increases the chances. Okay, so yeah. So initially, when you generate the sequences, you are just adding each monomer with a specific rate that was determined by experiments. So we took rates that were determined by experiments. Our collaborators did those experiments. And we took those rates, and there are rates of addition of each monomer, each of the 4 different types of monomers. And so when you do that, there's no specific energy minimization required. But the energy minimization aspect comes in when you are folding the sequences to create secondary structures. So for that, we used RNA fold, which is a well-known package for folding RNA sequences into secondary structures. Yeah, so there are no 3D structures. So we are making the assumption that a complex secondary structure can act as a proxy for biological function. So that seems to be a reasonable assumption to work with. Yeah. So any other questions for this first part before I move to the next? No, okay, good. So, so the conclusion of the first part is that ribozymes can be created via non-enzymatic processes, provided there are favorable environmental conditions like environmental cycling through these 3 different types of phases that I described. So the next question that we need to address is, it's not enough to produce long functional biopolymers. We need to encapsulate them in compartments. And not only do we need to encapsulate them in compartments, we need to find a mechanism by which these compartments, these sequences would be created inside those compartments, and a mechanism by which these compartments would grow and divide. And once they start growing and dividing, selection can step in, because then variants will be produced depending on the kind of components present in different compartments, and selection will act on those variants and preferably select ones which are more complex, the protocells which are more complex. So we need to get to the stage where these sequences are replicated rapidly inside compartments. And so so the reason for that is, this is just a cartoon that describes the process. So initial work done by Jain and Jostak showed that if you encapsulate RNA molecules inside lipid bilayers, vesicles formed out of fatty acids, then these RNA molecules start exerting pressure on the walls of the lipid vesicles, and eventually it creates an instability which makes them grow and eventually divide into two vesicles, and so on. So as the number of sequences inside a given protocell increases beyond a certain threshold, referred to as VT here, the protocell would divide into two, and the components of the protocell would become randomly distributed between the two daughter components. And once they are randomly distributed, if we start looking at a population of protocells, then the ones which have more complex phenotypes inside them would be preferentially selected over time. So this is the picture that we have in mind. And for that, what we need is a process of rapid replication of sequences inside protocells. So one way to establish that is through a process called a rolling circle mechanism. Now, there are many different mechanisms like the template-directed primer extension process that I mentioned. But if you take a linear template and try to extend a primer on a linear template and then try to separate it, that becomes a problem, because first of all, that requires very high temperatures. And secondly, as soon as you separate them, because there are a lot of complementary nucleotides that tend to just reattach itself back onto the template again. So in order to avoid this problem called the strand separation problem that is found for linear templates, we were motivated by work done by Tupper and Higgs and Christopherson et al. To look at a mechanism called the rolling circle mechanism, which is used in viruses. And the rolling circle mechanism assumes that there's a circular template here. A primer is attached to any portion of the template, and that gets extended, and eventually the strand which was getting extended can displace the strand at the other end and create this hanging portion. And eventually that hanging portion gets chopped off and creates an open-ended single-stranded RNA molecule. So this can, provided we can produce circular templates which are long enough, this can lead to a consistent way of replicating long sequences, which can also differ from the sequence of the template, because that's what we want, right? We want a variation to be produced, and that variation is produced because of the error-prone nature of the replication process. So what we did was we took a population of protocells, inserted circular templates in them, and started the replication machinery inside each protocell. We calculated the replication rate of a circular template of size 200 nucleotides. Well, forget about the details of the formula. Based on sequence level simulations. And so we can estimate the maximum possible replication rate here. And so once we did that, we started looking at different processes which could occur inside these protocells. So what are these different processes? So there's a process that can happen non-enzymatically, and also processes which can occur enzymatically once a replicase or a ribozyme is created inside these protocells. So you can have a single-stranded circular RNA getting converted into a double-stranded circular RNA like this, both non-enzymatically as well as enzymatically if there is a replicase to catalyze this primer extension process. A double-stranded can then get converted to an open-ended single-stranded RNA called L here. And this can happen both non-enzymatically as well as enzymatically, but the enzymatic rates are fast and so on. And open-ended single-stranded RNA can circularize under the action of a cyclase ribozyme and start creating new templates. So the key is to be able to produce a ribozyme which can act like a cyclase so that new templates are produced, and that will lead to increase in the number of replicates that are created. And maybe some of those replicates will start acting like a different type of ribozyme, which can increase the speed of replication and so on. So that's the idea behind increase in functional diversity over time as we increase the chances of replication of sequences inside these protocells. And there's also a degradation process that we took into account, which accounts for the fact that each of these types of sequences can degrade and break up into individual monomers. So the question that we wanted to address is whether there is a hierarchical emergence in functional complexity through emergence of different types of ribozymes through both non-enzymatic as well as enzymatic reactions. And if so, can we sort of guess what the earliest ribozymes could be? Can we put some ordering on the ribozymes which appeared right at the beginning? So what we did was to set up these equations. They look quite complicated, but what they are essentially saying is the rate of change in the number of a chemical species depends on the way in which that species can be produced from something else, the way in which it can get converted to something else, and the way in which it can degrade. So a competition between these processes lead to changes in the numbers of these different types of chemical species, single-stranded circular RNA, double-stranded circular RNA, open-ended single-stranded RNA, and these are ribozymes which are created during the process of replication. So we assume that if we can produce a large number of replicates through this template-directed rolling circle mechanism, then by chance, a ribozyme might emerge. And once that ribozyme emerges, if that ribozyme happens to be a replicase which can speed up subsequent replication reactions, then that leads to a positive feedback effect, which increases the number of sequences inside the protocell even more. And so we looked at 3 different types of ribozymes. So there's nothing sacrosanct about these 3. We are not claiming that these are the first 3 ribozymes that emerged. we don't have any evidence to say that. But for us, these three seemed natural to look at. So one is a cyclase which can take an open-ended RNA and circularize it to form a template. That was essential for us because the larger the number of templates, the more the replication process can occur. The second is the replicase, which speeds up the process of replication through this rolling circle mechanism. And we also looked at nucleotide synthase in situations where the environment was monomer deficient, because all these reactions require a constant supply of monomers. So in environments which are monomer deficient, you need monomers to be created. And so nucleotide synthase would do that. And we also looked at peptidyl transferase, so I'll come to that later on. So, so this was the game, essentially. We looked at replication inside a population of protocells through the rolling circle mechanism. And each replication event could potentially create a ribozyme or some other type of sequence. There was a small probability of creating a ribozyme from each replication event. And then we looked at the evolution of this protocell population by selecting protocells which have larger number of ribozymes and larger number of RNA strands in them. So in this case, we looked at just the larger number of RNA strands. So selection, so the fitter protocell variant would be one where there would be a large number of RNA strands compared to a variant which has a smaller number of RNA strands, which would be less fit. So number of RNA strands would be a measure of fitness in this situation. So what we found is that if you look at the probability of cyclase and replicase production, we found that evolution leads to protocells which contain both replicase and cyclase in amounts or in fractions which depends on the relative probability of the two. And if the most favorable situation is one where the replicase and cyclase formation probability is roughly similar, because then you find that the abundance of protocells which have both these types of ribozymes grow as large as 75%. Okay, so this is a mechanism of evolution towards increasing complexity, where complexity is measured by the diversity of different types of ribozymes contained in these protocells. So the reason behind this effect is sort of intuitive to understand. So if the probability of production of replicase was smaller than the probability of creation of a cyclase, then the number of cyclases created would be larger than the number of replicases. Then you would have a lot of these circular templates produced, but not enough replicase to act on those circular templates and enhance replication. On the other hand, if the probability of replicase production was larger than the probability of cyclase production, then a lot of replicases would be produced, but there would not be enough number of templates for them to act on. So both of these cause bottlenecks, which is avoided when these numbers are roughly same, which is why you see that the population of protocell evolves towards one where most of the protocells contain both a replicase and a cyclase. So that's the mechanism that we propose, which leads to increasing complexity of protocells. Yeah.
So why isn't the RC fraction changing?
Yeah.
First and second one, green curve remains close to 1/2 in both the cases.
Yeah.
And then it becomes 3/4.
So right.
Why isn't it changing?
So in the first two, so here this corresponds to the number of protocells containing R only, containing C only, and containing both R and C. And this is doesn't contain anything. Okay.
So it should be same as whichever is present.
Yeah. So since selection is acting on just the number and not on the type, the ones which have both R and C present will automatically have the larger number of strands in them because there are more templates to act on for replicases, and that speeds up the replication process and produces larger number of strands. So next, we looked at a situation where the environment is monomer deficient and introduced the possibility of nucleotide synthase emerging with a small probability during this process, along with a replicase and a cyclase. 5 minutes, okay, yeah. And we found similar results. There is a threshold value beyond which you don't have protocells containing all 3 types of ribozymes in monomer deficient environments. So unless nucleotide synthase is there to synthesize more nucleotides, the whole process breaks down, and we are back to protocells which are not very useful. So the whole idea of the game is to try to increase the number of useful components inside protocells, and because that will favor those protocells compared to others. So I'll skip this part and get to the next point very quickly. So the conclusion of this is evolution towards increasing functional diversity inside protocells is possible if different ribozymes act synergistically to enhance their copy numbers. And your ultimate aim is to increase the number of useful biomolecules inside the protocells and see whether protocells having larger number of functional biomolecules start dominating. And that's what we find from these simulations. So this still does not tell us how you can go from a world where you have only RNA, which played the role of both catalysis and as well as information storage, to RNA, DNA, protein world. So we haven't answered the question of how you can go to an RNA, DNA, protein world, but we have some evidence of how you can go to an intermediate world called an RNA, DNA world. And evidence of this comes from experiments recent experiments which are really mind-blowing in ways. So they found that there was a RNA polymerase which could replicate both DNA templates as well as RNA templates. So in other words, it could extend primers, both DNA primers on DNA and RNA templates and RNA primers on RNA and DNA templates. And these experiments mainly came from the group of Joyce at Scripps. And so we used their results to ask the question that what if you increase the space of possible reactions by introducing DNA nucleotides into the picture, in addition to RNA nucleotides? So you still don't have proteins, so you still need to rely on RNA enzymes like ribozymes, but you also have DNA nucleotides. So we found that the space of reactions can be expanded quite a lot when you include DNA nucleotides into the picture and allow for DNA sequences to be synthesized using these kinds of template-directed processes that Joyce and collaborators found. And one particular reaction that we found was very crucial was this autocatalytic reaction where the information about a replicase that is encoded in a DNA sequence is replicated by taking advantage by using the replicase itself as a catalyst. for this reaction. So this is an autocatalytic reaction that produces a replicase using a replicase and a DNA template encoding the replicase. And we could write down similar equations again based on the same spirit and introduce competitions between protocells where all these kinds of DNA sequences as well as RNA sequences are being produced, and found that the protocells which came to dominate the population and had the largest frequencies were the ones which contained both a ribozyme and the ribozyme encoding DNA sequence in it. And so this is a heat map which shows the abundance of protocells with both a replicase as well as a DNA template which encodes the replicase. And this is one pixel of that expanded here, the time evolution of that. And one interesting side effect that we see from these simulations is that the DNA encoding the enzyme appears at order 1. So it's roughly of order 1, whereas the replicase is, the number is more of the order of tens. So there are a larger number of replicases compared to the templates that encode the replicase. And the reason this is interesting is because this is what we expect in real living organisms, right? The living organisms have just one genome and many copies of proteins that are produced from those genomes. And so if you can think of this as the earliest form of a genome which has just one gene in it, so that single gene would act as a template which can be autocatalytically produced using the product of that gene itself. This sort of gives us hope that by expanding the space of possible reactions by including DNA nucleotides into the mixture, we can start thinking of getting to a stage where the earliest genome encoded by DNA sequences might emerge. Of course, initially it would, we are just looking at a single gene here. You have to make it more complicated than that. But hopefully this gives some insight into the transition from an RNA world to an RNA-DNA world where DNA sequences are encode heritable traits. So that's the story, and I'd like to end by, sorry to go over time by a few minutes, but end by acknowledging my collaborators. Most of the work was done by Shubham, who was a grad student who just completed his PhD and he's moving on to Sweden for his postdoc. Sudha was our experimental collaborator. We got all the reaction rates from her lab, and the work was done by Niraja, her grad student, and Julian was a friend and collaborator of both us, both me and Sudha. So we collaborated in the early part of the work. So thank you for your attention. I'll be happy to take questions. Yeah. We are, so in the first part, we were selecting for protocells which have higher RNA strands, higher number of RNA strands. So we are not looking at whether those strands are ribozymes or not. In the second part of the simulation, where we looked at RNA-DNA world, the example that I showed you, their selection is encoded by looking at, is imposed by looking at, by preferentially selecting protocells which have these ribozymes. Yeah.
My question was on the first part.
Yeah.
And the selection is for those which have higher sequences of, let's say, some RNA template.
Right.
And then you see emergence of cycles and replicase, and then kind of inflect the graph and higher top.
Right.
My question is, why would we be interested in selecting for those which larger number of those? Like, what is the selective advantage of the
Right.
has larger?
Yeah, so that goes back to the work of Chen and Jostak, who showed that if you have a large number of RNA strands inside a vesicle, it exerts some sort of osmotic pressure on the vesicle membrane and causes it to grow and divide. So this was a mechanism of coupling growth in the number of components inside the protocell and division of the protocell itself. So you have to understand that in the regime that we are talking about, the growth and division has to be brought about by a physical process, right? So the physical process involved is the osmotic pressure created by these RNA strands, and which causes it to grow and then subsequently divide. So there's no biological mechanism which can be invoked at that time, in that epoch.
All right. We could transition to the next talk, one more talk. But Supratim is around, so if you have more questions,